All right, so we are going to do chapter 3, and just as another reminder, once I go into presentation mode, I can't see the chat box, so please feel free to interrupt me. It's totally fine. Bust in if I'm going too fast or you have a question or anything like that. It's not rude, it is so I know you have a question. Okay, so here we are. We are on chapter 3 today, so we're going to be talking a lot about microscopes. All right, so some of the units of measurement for microscopes, we are looking at really, really small measurements. So we're getting into the micrometers, which is that little mu sign. So that it looks like a kind of fancy U. That's called a mu. And it's in your symbols. So when you're actually writing micrometers, you want to make sure you use a symbol, not a U. And nanometers, which is nm. So 1 micrometer is 10 to the minus 6th meter and 10 to the minus 3rd millimeters. So to put that into a little bit of perspective, millimeters are kind of equal to milliliters, which would be cc's. So if you're giving somebody some cough medicine, because I've been sick, and it says 5 milliliters, that's a volume, right? But it's pretty small, but it's kind of measurable. You can see it. So it's kind of equated to millimeters, which would be a length rather than a volume. It's, pretty small. And then we're going into micrometers. So yeah, you can't see that. Well, you technically can see 1 micrometer. If you pipette it up, you can see just the teeny tiny little bit that's in there. You cannot see 1 nanometer. So when you get down to 1 nanometer, that's 10 to the negative 9th meter, which is 10 to the negative 6th millimeters. And then we just have a couple other conversions on this slide. Oops, somebody's coming in. Let me admit them. There we go. And then we have a couple other conversions here where we have 1,000 nanometers is 1 micrometer, or flip that around, 0.001 micrometer is 1 nanometer. So it's not exactly math, it's just a bunch of just moving those zeros around. What we really want to say is that we're talking about something very, 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 very small. Most cannot be seen without a microscope. And even if you can see it, like you can see a tick there in this picture that they're showing, they're very small. And the things that are going to infect you are even smaller because they're inside that tick, a parasite, right? Okay, so we did look at this microscope on Monday, which was Anton van Leeuwenhoek's crazy little machine. This is a simple microscope. It has only 1 lens. So it's similar to a magnifying glass, but with a really a higher magnification lens. So of course, when he made his lenses, he kept that information pretty patented and secret. And he just had a simple microscope. And that's why it took us so long, I think, to really move into the world of microbiology, because this is not a very efficient microscope. Well, what we use for a lot of microscopy is light microscopy. And this is any kind of microscope that's going to use visible light to observe specimens. And we have a number of different types of light microscopy. There's compound light microscopy, dark field microscopy, and we're going to go through each one of these. There's a slide on each one, so we're just not going to explain them on this slide. Phase contrast microscopy, differential interference contrast or DIC microscopy, and fluorescence microscopy. I might have confocal on this slide too. I did, yep, and confocal microscopy. So all of these are different types of light microscopy. They're going to use some form of visible light. Now, when we get down into fluorescence and confocal, it's a little bit of a trick with that one because we're using a light, a laser that you cannot see, UV light, and it's going to hit your specimen. But what it does to the specimen is that it creates a fluorophore-2 glow, and that gives off a light microscopy. That's why those two are grouped in the light microscopy. So let's take a look at all of these different types of light microscopy. So compound light microscopy, this is the most common type. This is the microscope that you see usually in labs. You might see on TV. For some reason, they always, not always, but a lot of television shows use like one lens, which is weird. I don't know why you would just look through one eye. So I was always taught open both eyes, look through, adjust them as needed. Anyway, a compound microscope is going to have more than one lens. So the image from the objective lens is going to be magnified again by the ocular lens. The ocular lens is the one closest to your eyes. The objective lens will be the lens down at the bottom. And in this case, there's 4 of them on this particular microscope. This is kind of like the microscope we actually have in our lab. Your total magnification will be your objective lens times your ocular lens. On the lab site, which I will send an email about, and I will, I'll send an email and I can talk more about it at the end of the lecture. I have put up some videos for microscopy, and they do have one. It's kind of boring. I'm sorry. It is from, oh gosh, it's Oregon State, on how to use one of these microscopes. And they do it very slowly. They walk you through each one of the objective lenses. But since we can't really do anything in person quite yet with a big class, this is where we're at. So anyway, each one of those objectives would be times your ocular lens to get your total magnification. Now, those objective lenses range from 40 to 100 and everything in between. I think it's 440, I want to say 80, and then 100. And 100 is your oil immersion, which we have on a further slide. Your ocular lens is always 10. So it would be 10 times 4 for your smallest objective lens for a total of 40. 10 times 40 for the next objective, obviously 400. So I'm not going to do math for you, but get the idea. So in compound light microscopy, we have a couple definitions here. Resolution is the ability of the lenses to distinguish 2 points. And a microscope with a resolving power of 0.4 nanometers can distinguish between 2 points that are at least 0.4 nanometers apart. So it's from one end of the cell to the other. A shorter wavelength of light will provide a greater resolution. So long wavelengths, it's just like a wave, literally a wave like on our lake. You've got the big ones, and then you have ones that are very short and quick. Those shorter ones don't have as much time to be lost. So the light is going to actually hit your specimen. When you have a long wavelength, you're going to lose some of your light. So you lose some of your resolution. The refractive index is a measure of the light bending ability of a medium. So it's hard to imagine because we live in it, but this air that we're breathing is a medium. So just like fish in the water probably don't think about that they're in a medium of water, we are in a medium of air. So when light comes through, when light is coming from the sun, right, it's going to bend when it hits the earth, and it's going to bend when it hits air. It's changing its direction, just like when you put a stick in water and you look down, and it doesn't look straight. It looks like it's bent. That's actually the light bending because of the medium. So when we're using a microscope, we're looking at very, very small things. If light bends too far away, we're not going to see our specimen because the light isn't going to come through the objective lens. So the light may refract after passing through a specimen to an extent that it does not pass through the objective lens. And this is going to happen when we get to an objective of 100. So when you have a 100, your light is bending away, and that's what's showing here, that red light. It can't get into the objective. So what we do is we use what's called immersion oil, and that's going to keep the light from refracting. It basically catches the air. This is physics, really. It catches the light, no, I said catch air, catches the light and directs it through the objective lens. Sorry. Eventually I will no longer have a cold. Okay, so this is what it looks like, and this is why we use oil on our highest objective lens on the compound microscope. Okay, so some of these different types of compound light microscopy include bright field illumination. In bright field illumination, dark objects are visible against a bright background. Light that's reflected off of the specimen does not enter the objective lens. Whoops. So basically what this says is that it's illuminating you're seeing a shadow. Let's put it that way. That's a better way to say it. So you're seeing a shadow of a specimen. So that's why we can see the cilia in this particular picture, and you can see there's some vacuoles inside that look granulated. We're essentially seeing a shadow. So now this is fun. I've got a little video for you guys to watch. And then I'm going to see if I can make these available for you. So somebody else is going to talk to you about this.
Light microscopes are useful for studying specimens ranging in size from about 1 millimeter to about 200 nanometers. It's impossible to study anything smaller by light microscopy because the shortest wavelength of visible light is about 390 nanometers, and anything smaller will not interact with the light. The most common form of light microscopy is called bright field microscopy. In this form of light microscopy, the light beam passes through the sample and into the objective lens. Let's look at the parts of a light microscope to see how a specimen is magnified and viewed. The lamp is where the light beam is initiated. This beam is divergent, spreading out as it approaches the condenser lens. The condenser lens gathers the beam of light and focuses it so that it converges on the specimen with high intensity in a small area. The specimen usually last slide that was on the light path away from the specimen. After the beam leaves the specimen, it passes through the objective lens. The objective lens performs the bulk of the magnification of the specimen. The eyepiece receives the light beam from the objective lens and refocuses it to recreate the image in the viewer's eye. Lenses found within the eyepiece are called ocular lenses.
Okay. So that is a little video that I will try to put up separately onto Brightspace for you. Okay. Yes, so dark field microscopy now. In dark field microscopy, light objects are visible against a dark background. So you can see the difference in this picture versus the bright field where we saw just the shadows of the image. In here, we have an opaque disc, which is placed into the condenser. And then only light that's reflected off of the specimen will enter the objective lens. So now those features that we saw in the previous figure are illuminated. And so we see those little, they were dark vacuoles. Now we see them as little white bright spots. Phase contrast microscopy. Now, phase contrast microscopy will allow for examination of living organisms and internal cell structures. So this is important to say living organisms. By using phase contrast microscopy, you're not killing your specimen. You're not fixing it. You're not staining it. It's still alive, it's moving around, and you can see quite a bit of detail. So again, looking at that picture, we can see even more detail than we did in the previous pictures. And this is because it's bringing together two sets of light rays, direct rays and diffracted rays, to form an image. So by using both of these types of lights, we're able to visualize even more of the internal structure. And you can kind of see the cilia on the outside there. You can see the vacuoles, but you can see everything in a little bit more detail. So now we want to see even more detail. We're going to use differential interference contrast, or DIC microscopy. It's similar to the phase contrast microscopy. It's going to use two light beams. It's also going to split those. Oops, somebody's coming in. Let me admit them. Okay. So it's like phase contrast, we're using two light beams, but we're going to have prisms inside of the microscope that are going to split those light beams, and it's going to give even more contrast and color to the specimen. So the more that we're going to refract that light and direct it at the specimen, you're hitting it, bombarding it with more than one type of light, those are going to illuminate different structures. And as you can see, this final picture here is really illuminated. You can see a lot in there. Now those little vacuoles that we saw are really defined. They're larger, and we can actually see there's even small dots, small vacuoles. Those could be chloroplasts even. They kind of look like that's what they're holding. All right, so on to fluorescence microscopy. Fluorescence microscopy uses UV, which has a very short wavelength of light. We can't see UV light. So why is it under the light microscopy? Because that UV is going to hit your specimen and it's going to illuminate a visible light for you. So the fluorescent substances that are in our specimen will absorb the UV light and emit longer wavelength or visible light. So cells may be stained with fluorescent dyes, fluorochromes, if they do not naturally fluoresce. So there are some things out there that will naturally fluoresce, some types of algae and such, but generally speaking, most of the things that you're going to want to visualize are going to have to be stained. So that means that the cell or the specimen will be fixed, it will be killed, and it will be stuck onto a slide. So the fluorescence microscope that we have in our lab is, I didn't take the actual picture of it, but it is an EVO cell imaging system. This is what it looks like. It has a very small footprint. You just plop it on a desk. And then these are some cells, some fat cells that were differentiated from Wendy and her student Courtney. And they did this back in 2019 before everything shut down. I believe it's the summer of 2019. And so you can really see a lot of detail in those cells. We're looking at where you see the blue. Those are that's a nucleus. We almost always stain our nucleus with blue. It's called a DAPI stain. And then you can see the little fat globules, and those are in green. And then you can also sort of see some cytoskeleton in sort of an orange, where it's stretching out, where the cells are kind of touching together. So that's just a beautiful image that we were able to get from our own fluorescence microscope.
That's awesome.
It is pretty cool. And we don't, so this imaging system, the fluorescent microscopes that we used to have actually had ocular sight as well. We don't do that anymore. Everything is digital. So there is a really old fluorescence microscope hiding in our lab behind a dark field door. That door is going to go away. That microscope is going to go away because everything is just this beautiful little system now. So confocal microscopy. Now this is the next step of fluorescence microscopy. So we still stain our cells with the fluorochrome dyes. And then we use a short wavelength light to excite a single plane of a specimen. And then each plane in a specimen is illuminated and a three-dimensional image is constructed with a computer. So we have to, it's so easy to forget when we're working on computers, we're looking at each other on a computer, we're looking at our images on a computer. It's hard to forget we live in a three-dimensional world. We don't live in a two-dimensional world. So when you look at these cells, you have to remember they're not flat. They actually have depth to them. So that's what confocal microscopy gives us, the ability to see the depth. And so it's going to basically slice it, take images of slices, and then build them together into a three-dimensional image. And they can, confocal microscopy can examine layers of cells to a depth of 100 micrometers. I don't believe that our fluorescence microscope at the moment has confocal on it, but it can have that. So we can have that added in the future. Okay. So two-photon microscopy. In this one, it's just a step up. The cells are still stained with fluorochrome dyes. And then we're going to use 2 photons of long wavelength light to excite the dyes. Now this one can study living cells up to 1 millimeter deep. So if you have a cell that can tolerate a stain, you can do living cells with 2 photon. You're not going to be bombarding it with a UV light. You're not going to be killing it necessarily. This would be particularly useful for if you needed to see cell division over real time. A lab that I used to work at, some of the people that were doing this, they were actually looking at fungal cells, and they just used some green fluorescence, and they were watching them divide to see how that process was going on. But they only live for about 24 hours because after a while, the microscope will kill them. Super resolution light microscopy. In super resolution, we're going to be using laser beams. One wavelength will stimulate the fluorescent molecules to glow.
Was that Dr. Evil?
Yeah. All I wanted were sharks with freaking laser beams. Okay.
Nice.
That's a great show. The second wavelength will cancel out all the fluorescence except for any fluorescence that's down to 1 nanometer. So in this case, what we're doing is we're kind of competing the beams out to get rid of the extra noise and to focus it down. So we stimulate it to glow, and then another laser comes in and it cancels out any of the extra light so that we can have very precise resolution. And again, resolution is the distance between one side to the other of a cell or an organelle. And then this computer will scan the specimen nanometer by nanometer and put the image together. So this is all done digitally again. Okay, I think that's it. This is an interesting technique that has not changed very much. It is not a light microscopy, and it's used more in material science, I'm going to say, than looking at microbes anymore. It might look at biofilms. So scanning acoustic microscopy is using the same technique that dolphins use to find things in the water, that we use sonar. It's the same idea. It's just very, very, very small. So it's going to measure sound waves that are reflected back from a specimen. It can be used to study cells attached to surfaces, so like a biofilm. It can have a resolution of up to 1 micrometer, but what we'll really see for scanning acoustic is going to be in material sciences. So when we're trying to figure out, is this a really great material that I can use for cell phone building or whatever? I'm not a material science, obviously. And they want to see how many bumps it has on the surface and if it can hold more data or more digital capacities. So I haven't seen it used in probably, it's really interesting, but I don't think we've used this technique very much in probably the past 10 years for real microscopy of cells. Now this, on the other hand, is a microscopy that is growing, electron microscopy. It uses electrons instead of light. So that's interesting right there. We're going to bombard our sample with electrons. The shorter wavelength of electrons will give a greater resolution. And it's used for images that are too small to be seen with light microscopes, and this is such as viruses. So we're looking at a paper from Dillard et al. In 2019, and they have a grid that they have captured some virus-like particles. And these are HIV virus-like particles, and they are being imaged by a bombardment of electrons. So this is on an atomic level because, right, everybody in here has taken chemistry probably with Clay. You know what an atom is, and you know the electrons are outside of the atom. We literally rip those electrons off of there. You can do this using a copper anode, and then you bombard your sample with those, and what those electrons are going to do is they're going to rebuild on that atomic level of an image for you. So this is 1 micrometer, this particular picture that you're looking at, and B is really the better visualization. So you see those 4 circles, those are not the virus-like particles. Those are just part of the media from which they're putting the virus-like particles onto. The littler dots, those are your virus-like particles. So there's a couple different types of electron microscopy. We have transmission electron microscopy. And that is where we have a beam of electrons that pass through an ultra-thin section of a specimen, then through an electromagnetic lens, and then it's focused onto a projector lens. I think there's another, yes. Specimens may be stained with heavy metal salts for contrast. Very often they use a little bit of gold in there. And so these specimens, it takes longer to prepare the specimen than it does to visualize it. And in some ways, it's not really microscopy because you're not, now there is an eyepiece, but you're not actually going to be able to visualize your what you're looking at. You can see the specimen itself that you've prepared, but what you're trying to see is too small to be seen through a viewing eyepiece, and it has to be rebuilt by the computer. So these electrons that are bombarding, like I said, we're on an atomic level, and that's going to be rebuilt. So this is, like I said, I mean, very, very, very small, right? It's going to magnify objects 10,000 to 10 million times the resolution of 10 picometers. So super, super small. So small, what you view through your eyepiece would be the sample, the slide and that sort of thing. So you know you're kind of lining it up. But what you're actually going to see is going to be built on a computer on an atomic level. And there's a lot of work that has to be done with that, both by the technician who's working the microscope and the computer. You can't just plop it on and turn a button and you could see it. It's really a lot more complicated than that. Now, in scanning electron microscopy, we have an electron gun that produces a beam of electrons that's going to scan the surface of an entire specimen. Secondary electrons emitted from the specimen will produce a three-dimensional image. So scanning electron microscopy in some ways is like your confocal fluorescence. In this case, we are rebuilding a three-dimensional structure. This is sort of similar to what we do for protein crystallization. You know, when we're trying to determine the three-dimensional structure of a protein, we're using a type of scanning electrons where we bombard our sample and we move our sample around, and we can get a three-dimensional image rebuilt by the computer and through lots of work. I used to be a structural biologist. That's what I did, and I'd like to do that again. Did that start at SKC. But it's a lot of work making sure that your programming is correct, that you're not making up your image. Let's put it that way. So this can magnify objects 1,000 to 500,000 times with a resolution of 10 nanometers. Anything else? I think we're good. Okay, so now somebody else is going to talk to you from the book about this electron microscopy.
Electron microscopes use electrons to probe a sample. Electron microscopes are useful for studying samples in the size range of 10 nanometers to 100 micrometers. This includes specimens as small as ribosomes, up to specimens as large as red blood cells. Transmission electron microscopes, such as the one shown here, are used to produce two-dimensional images of internal structures of cells, while scanning electron microscopes are used to produce three-dimensional images of the surface of specimens. Electron-dense compounds, such as heavy metal salts, can be used to stain specimens for electron microscopy, increasing the contrast and resolution. Let's look at the parts of a transmission electron microscope to see how a specimen is magnified and viewed. The electron gun initiates a beam of electrons. This beam is divergent, spreading out as it approaches the condenser lens. The condenser lens gathers the beam of electrons and focuses it so that it converges on the specimen with high intensity in a small area. The condenser lens in an electron microscope is electromagnetic. The electron beam next passes through the specimen. In a transmission electron microscope, the specimen is a tiny slice of cells that rests on an equally tiny copper grid. This grid is inserted into the microscope at the location shown here. When the electrons interact with the sample, they are either reflected and refracted or absorbed by the sample. The beam then continues to diverge downward on its path away from the specimen. After the beam leaves the specimen, it passes through the objective lens. The objective lens performs the bulk of the magnification of the specimen. The projector lens receives the electron beam from the objective lens and refocuses it to recreate the image that the viewer sees in the fluorescent screen. An internal camera is used to photograph the specimen. You may have noticed that these two types of microscopes share similar parts. Both have a source of light or electrons, a condenser lens to focus this source, and an objective lens to refocus the image. However, you may also have noticed that the path of light is from bottom to top in the light microscope, while the path of electrons is from top to bottom in the electron microscope. All types of microscopy are concerned with resolution. The resolution of an image is the ability to distinguish between objects that are close together. Resolution depends on the wavelength of the source, with smaller wavelengths leading to higher resolution. The average wavelength of white light is 550 nanometers, while the wavelength of electrons in the electron microscope is 0.01 nanometers. This is why electron microscopes are able to resolve objects much smaller than light microscopes.
Okay. So again, I'll try to make these available to you. They're kind of a nice little review, and you don't have to listen to my voice all the time. Okay, so let's look at a few different final things, and then we're going to get into some staining. So scanning tunneling microscopy. In scanning tunneling microscopy, we use a probe, and it's a tungsten probe, to scan a specimen, and it's going to reveal details of its surface. So it has a pretty high resolution of 1/100th of an atom. That's pretty insane. Why would you want to see something like that? I'm kind of guessing that maybe you guys are thinking, why would we want to see what something looks like that's that small? Well, often these techniques are used in conjunction with structural biology. But what we want to do is, if you can see the outside of a cell, such as a virus, we can determine, say, the viral spike that might be on there, its exact shape. And then we can go and develop antivirals to such a thing. We can work on developing antibodies and also, of course, vaccine development. So once you can visualize these things, you are able to use a number of other techniques to cure disease or to, cure disease is pretty much, I think, what we'd want to do. Trying to think. You can use some other biotechnology, but let's go with curing. Atomic force microscopy uses a metal and diamond probe. This is placed into the specimen, and then the movements are recorded. And this is going to give you three-dimensional images at a near atomic detail. So again, this is really looking at that detail, but we can get into the cell. So maybe you really want to see the detail of an organelle within a specimen. Okay, so as staining is our next step. So we talked a lot about the microscopes, but often we're going to want to stain it so that we can then use the microscope. We talked a little bit about that when we were talking about the fluorescence, but there's a lot of different types of staining. And in staining, we're going to color the microorganisms with a dye that's going to emphasize certain structures. The first thing that you want to do is make a thin film of the material containing microorganisms, and that's a smear. So when we do staining in the lab, often everybody, because you forget how small these things are, the first smear everyone does is usually way too thick, and then I have to, maneuver things around so that they can actually see their individual cells. We go to the very edge of the smear usually. And that's because naturally you think, oh, this can't be enough, and you smear it onto your slide and it's big chunk. And you forget that's hundreds of thousands of cells that are there. Microorganisms are then fixed to the slide, and that's going to kill the microorganisms. So the way that we fix bacteria very often is just using heat. It depends on the type of stain that you're going to do. Now, if you're going to do a fluorescent stain, like those cells that we saw earlier that Wendy and Courtney did, that's a different type of fixing, and it's going to use a type of, it's kind of like using a formaldehyde type fixer. And it kills the cells, but it also has them adhere pretty tightly to the slide or even the well of the plate that you're using. Live or unstained specimens have very little contrast with the surrounding medium. So if you ever want to look at a live specimen, you're doing that to see the cell behavior, to see how they divide, or how they interact, or to see if these bacteria use a lot of contact with one another, or are they going to build a biofilm, that sort of thing. But you're not going to get a lot of detail. So really, most of the time, you're going to want to kill your organism and stain it in order to really visualize it. And you cannot use live specimens at all. You're not really going to see anything with viruses at all. You could see the cell behavior of the cell that it infected. So if that's something that you wanted to see, you could use a live specimen. Sometimes one of the ways that we know a differentiated cell that has a viral infection in it, they'll pile together, whereas the uninfected cells will stay flat on a surface. So that might be something that you want to visualize with a live cell, but you cannot see a virus unless you stain it and use electron microscopy. Okay, so we're going to do this video.
Microscopes are very useful tools to study specimens smaller than about 1 millimeter. Light microscopes and electron microscopes use lenses to focus light or electrons passed through a specimen to produce a magnified image. Both light and electron microscopists utilize stain to increase the contrast and visibility of the specimen under study, as well as to aid in the classification of these specimens.
Okay. And so there you can see right there an unstained specimen. I know it's just a drawing. Sorry, just trying to catch up on emails while I'm doing this. And then once you stain it, though, you can really see the differences. And so let's look at some of these different types of stains. Stains will consist of a positive and a negative ion, one of which is colored, and that's the chromophore. And a basic dye, so remember basic is going to be above the pH of 7. So it's a base. The chromophore is a cation. And then in an acidic dye, the chromophore is an anion. So when we're acidic, below the pH of 7. Sometimes it's useful to stain the background instead of the cell, and that's called negative staining. That might be very useful if you are trying to see the outside of a cell and you want to, again, it's kind of like looking at the shadow of a cell, that sort of idea. And you really want to see, the flagella or the cilia or something that's outside of the cell. So simple stains. When we do a simple stain, we use a single basic dye that's going to highlight the entire microorganism to visualize cell shapes and structures. A mordant may be used to hold the stain or coat the specimen to enlarge it. Okay, I guess this guy goes on.
Light microscopists utilize stains to increase the contrast and visibility of the specimen, as well as to learn more about the features of the specimen. There are 3 main categories of stains used by microbiologists: simple stains, differential stains, and special stains. Simple stains are made of a single basic dye that interacts with the biological sample and stains it. After treatment with the dye, the sample is rinsed with water, leaving the stained sample. Simple dyes are used to determine size, shape, and arrangement of cells. Crystal violet is a purple stain that stains most bacteria uniformly. Safranin is a red dye that stains most bacteria pink or light red. Methylene blue is another basic dye that stains most bacteria a light blue color. Some of these dyes are also used in more complex staining techniques, such as differential stains. Most of the stains used in microbiology are differential stains. Differential stains use more than one type of dye and can be used to distinguish between different structures. The gram stain is the most frequently used differential stain. It's used to distinguish between gram-positive cells, which have a thick outer cell wall composed of peptidoglycan, and gram-negative cells, which have a thin outer cell wall. The procedure of gram staining involves 7 steps, during which the samples are stained with crystal violet dye, treated with a mordant, rinsed, treated with a decolorizing agent, and counterstained with safranin. Gram-positive cells are stained a deep purple color from the crystal violet dye, but gram-negative cells are decolorized and then stained pink by the safranin. The acid-fast stain is a useful differential stain because it only interacts strongly with a subset of bacteria in the genus Mycobacterium that have a waxy material in their cell walls, as well as the pathogenic strains of the genus Nocardia. The acid-fast staining method involves several steps, during which the samples are stained with carbofusion while being heated over steam, cooled, decolorized with hydrochloric acid, and counterstained with a simple dye such as methylene blue. Some bacteria produce endospores. Endospores form inside the cytoplasm of the bacteria and are highly resistant to desiccation, heat, and radiation. Their cell walls are impenetrable to most chemicals. The endospore stain uses heat to drive the dye malachite green into the endospores. After heating with malachite green, the slide is rinsed with water to remove the dye. The slide is then counterstained with safranin. In the final sample, vegetative cells are pink and endospores appear green. The endospore stain is a useful tool in classifying bacteria. Special stains are stains that interact with special bacterial features or structures such as the flagella or capsules formed by certain bacteria. Negative stains stain the background, leaving cells colorless. These acidic dyes are repelled by the negative charges on the surfaces of cells. Examples of negative stains include eosin and nigrosin. Some bacteria use tiny structures called flagella for locomotion. Flagella are too small to be seen with a microscope, but they can be stained in a multi-step process involving the dyes pararosaniline and carbofusion, and the mordants tannic acid and potassium alum. The number and arrangement of flagella are important in the classification of bacteria.
All right. So, it's a little boring to watch these videos, but I think that really shows all the different steps of the stains and how important they are. We're going to go through some of these again. So the differential stain, it's used to distinguish between different bacteria where we have gram-positive and gram-negative. And also the acid-fast stain that they went through. So this figure that we're looking at, that's 313A in the new book, but again, you can just use this figure from the PowerPoint here, shows the different steps of gram staining. And as that video sort of went through, you start with crystal violet, everything turns purple. You have a mordant, which is going to fix that crystal violet into the peptidoglycan. You decolorize it with alcohol. So if you don't have a thick layer of peptidoglycan, it's going to wash off in saffron and will counter stain. So if it's a gram positive, it'll stay purple. If it's a gram negative, it's going to turn red. And so here, this classification will get bacteria to gram positive or gram negative. All bacteria really fall into these two groups if they have the peptidoglycan in their wall. So gram positive bacteria have thick peptidoglycan cell walls, and gram negative bacteria have thin peptidoglycan cell walls and a layer of liposaccharides. And so when we are looking at treating a disease with an antibiotic, we need to know if it's gram positive or gram negative right off the bat. Most most antibiotics that we have are designed to degrade peptidoglycan. So they target thick peptidoglycan cell walls. So most of our antibiotics that we have in our toolbox are going to go after gram positives. So this is penicillin, any penicillin derivative, and quite a few others that were developed because of penicillin allergic reactions. They're all against gram positive. We have very few gram negative antibiotics out there, and this is because gram negatives, you have to target something else in that cell. You can't target the peptidoglycan cell wall because it's so thin, and it has basically a slippery surface. It's got a lot of lipopolysaccharides. So lipo is for fats, right? And so because these cells are slippery, they're hard to target with antibiotics. We do have antibiotics against gram negatives, but generally speaking, if it's determined you have a bacterial infection, the first thing people will do is give you that gram positive antibiotic. And then if it's determined that you have a gram negative infection, then they'll use the other antibiotic. But we don't really want to overuse those gram negative antibiotics because there's very few of them, and we don't want to develop antibacterial resistance. So the acid fast. Acid fast will bind only to bacteria that have this waxy material in their cell wall. It's a muric acid. It's not decolorized by acid alcohol. And so it's used for the identification of the specific bacteria that will cause respiratory illnesses such as tuberculosis. So this is Mycobacterium, and it also will color nocardia. And so this is a little table down there, shows what you're going to see at each step. If you have an acid fast cell, it's going to stay red. If you have a non-acid fast cell, it's going to turn blue. So when we look at our picture, we can see we've got some M. tuberculosis, Mycobacterium tuberculosis in there. And then there's also some blue. And so those are cells that are not the tuberculosis cells. So we have a mixed population. And then finally, our special stains. These special stains are used to distinguish different parts of microorganisms, such as a capsule, an endospore, or the flagella. So we'll look at each one individually on the next slides here. The capsules. So we do a negative stain for capsules. So because capsules are going to be on the outside of a cell, we want to do a negative stain to see where an absence of stain indicates a capsule. So capsules are gelatinous covering that do not accept most dyes. So what we can do is a suspension of India ink or nigrosin, and that's going to contrast the background with the capsule, which will then appear as a halo around the cell. So why this might be important is that, so as they actually showed in that video, I don't know if you caught it, but we were looking at E. coli that was pathogenic and E. coli that was non-pathogenic. I think it was the other way around. I had the non-pathogenic on the left side. But the non-pathogenic E. coli do not produce capsules. So while all E. coli is going to gram stain as a gram-negative rod. So it's going to be a red little rod. If you want to know the difference between a pathogenic and a non-pathogenic, so say you are working in a lab and you've been getting some specimens from, I don't know, from some romaine lettuce that is making people sick, and they say, why are people getting sick? And so you go in and you're staining, you go, oh, I've got E. coli on here, but is it pathogenic or not? And then you stain it with your negative stain for capsules. You find there's a capsule, you go, it's E. coli that's causing it. Or you say, no, it's not causing the disease. We're going to have to look for another bacteria such as Salmonella. So that's what's useful with these capsule stains. Endospore staining. So endospores are super tough. Oh, these guys are pretty neat. Actually, you can send an endospore into space, leave it on the moon, which has, well, okay, not on the moon, but they left it on a satellite for quite some time. And then you can bring it back to Earth or bring it into the space station and you can bring it back to life. They are so tough. So endospores are resistant, dormant structures inside some cells, and they cannot be stained by ordinary methods. So we're going to have to look for a different method to see if these cells are producing endospores. The primary stain for an endospore is the malachite green, and you have to use heat to fix it on there. You can decolorize just using water, and then counterstain with safranin. And so what we're looking at in this picture, we see the safranin has stained the vegetative or live cells with red, and anything that is in a dormant state will appear green. We've also found these endospores, in permafrost, where it's been covered with a glacier for a really long time, and the glacier retreats and there's some permafrost there, and you get these endospores and you can bring these bacteria that are thousands and thousands of years old back to life. They're absolutely fascinating. Oh, so there we are. The spores will appear green and they'll be within the vegetative cells that are red or pink. And then I believe this is our final, flagella staining. So flagella are structures of locomotion. There's different types of flagella, which we'll go into in a later chapter. You can have them all around. You can have them at the ends. You can have one. You can have two. And so E. coli actually have a flagella at one end. And that flagella is often indicative of pathogenesis as well. So the flagella staining uses a mordant and carbonyl fusion to thicken the appearance of the flagella, and then it makes them visible under the light microscope. Flagella are super thin structures that you can't see without stains. You might see a specimen move if you've got a live one, and then you suspect that it has a flagella, but then you might want to do your stain to be sure. Did I see one little flagella or did I see several? And by counting those or knowing where their position is, you can identify the type of microbe that you have. All right, so in summary, we've got all of our different types of microscopes. We can visualize microorganisms using light microscopy, and the different types are compound, dark field, phase contrast, DIC, and fluorescence. Scanning acoustic microscopy is a rarely used one, but it is used for biofilms, and it's kind of a neat technique. It's just not used as much. Electron microscopy has a number of different types. So we have transmission electron microscopy and scanning electron microscopy, where the scanning electron microscopy will give you a three-dimensional structure. And then atomic force will give you really fine detail of the outside of a cell, or if you use a probe to go into the cell, of an organelle. And then staining is super important for most of the types of microscopy that we use to emphasize certain structures. And then the different types of stains we've got, well, I didn't put simple on here, but we do have simple stains, We have gram stains, acid fast, and our special stains. So I think that is the end. We are a little bit early, so we're at 10:36. So I'm going to stop presenting here. And before I open it up for questions, I just want to repeat that I have put up some videos and some work to do in the BIOS 131, which is the laboratory section of this class. We are doing a virtual lab that is on your own pace. So I'm going to track and make sure that everybody is doing some of this work. And then I'll put up the next module. So this is all on microscopes and staining that will be for the lab. And then we'll come up with some new stuff and hopefully some fun at-home labs. All right, I'm going to stop recording here and open it up for questioning.